Okay, good morning everyone, good morning. Good morning all. Good morning. Okay, good morning. Let's continue. So in the yesterday class we have finished the concept of transport in plants, right? So we are left with nitrogen metabolism, secondary metabolites, then followed by the sensory photobiology chapter. Okay, let's quickly understand nitrogen metabolism. Good morning everyone. So friends, what kind of questions we can expect in this particular nitrogen metabolism? So you can consider this topic also very important topics, because out of 10 question papers the question was appeared 6 to 7 times, and most of the time you can expect a kind of a straightforward or a direct question only. Mostly friends, mostly you can get the question from the statements only, right? What you people has to read exactly in this particular topic is, friends, you can expect the question from the processes. Okay, you can expect the question from the genes. Okay, and most importantly friends, you can expect the question from the bacteria, or you can expect the question from the enzymes. Okay, these are all the only ways that you can expect the question from this particular nitrogen metabolism topic. Okay, beyond these four ways friends, there is no other way of asking the question. Okay, so make sure that if you look into the 2022 September paper, we got a simple 2 marker question, right? Simple 2 marks question. And if you look into the 2022 June paper, I mean to say February paper, we did not get the question. But if you look into the 2020 November paper, we got 4 marker question. If you look into the 2019 in the June also we got the question, December also we got the question, both time we got the question for the 4 marker. So either part B or part C we can expect the question. So that's the reason this topic seems to be a small topic, but it is your responsibility to read the entire topic very correctly. Okay, now let's start nitrogen metabolism. So no friends, along with the cycle we have to understand the metabolism as well. And we all know very well, when compared to the oxygen as well as carbon dioxide, how much amount of the nitrogen is present in the atmosphere, friends? So we all know that 78% of the nitrogen is present in the atmosphere. And the point is here, friends, we people, we animals as well as plants can't survive without nitrogen. Why the plant and animal cannot survive with nitrogen? Because nitrogen is the one that plays a crucial role in the production of amino acid, in the production of a nucleic acid, in the production of a protein. That is the reason what we can say that nitrogen is extremely important for the plant as well as the animal. But unfortunately, understand every one of you, Whatever the nitrogen that is present in the atmosphere, what do we call it as, friends? We call that particular nitrogen as the chemically inert form. Friends, what do you mean by the word chemically inert form? That's a valid point. Chemically inert, or in a simple words, nitrogen present in the atmosphere, it is in the N triple bond N. If you look into a plant, or if you look into the animal or plant do not have any kind of a enzymes to break this particular N triple bond N. That is the reason animal and plant can't use the atmospheric nitrogen. But understand my points correctly. Plant and animal can't break down the nitrogen, but plant and animal can't survive with nitrogen. So now what is the ultimate goal? The ultimate goal is that someone has to break down the nitrogen into the most simple forms. What are the most simple forms of the nitrogen, friends? NO2, NO3, NH3 and NH4 plus. So when someone convert, remember my words correctly, when someone convert the nitrogen into the most simple forms, we plants and animals can easily utilize it. So once, who will fix this particular nitrogen into simple forms? Of course, the bacteria. Bacteria fix in the soil. From the soil, who will take, friends? From the soil, plant will take. Up from the plant, who will take? Animal will take. So what is happening? Try to understand the concept. A little amount of the nitrogen, it is keep on circulating and recirculating. The process by which limited amount of the nitrogen is circulated and recirculated through the world of living organisms. That is what exactly, friends, we will be calling it as the nitrogen cycle. Understand the point. From one tropic level to the another tropic level, nitrogen is continuously circulating. You know, the nitrogen cycle never be stopped, because every organism, in order to produce the proteins, amino acid, they will be requiring a continuous supply of the nitrogen. That's why the nitrogen cycle will be circulated and recirculated. And don't think that vast amount of the nitrogen is circulating and recirculating. Every single time, friends, a very limited amount of the nitrogen will be circulated as well as the recirculated. Now all of you pay attention. Nitrogen cycle will be occurred in a following steps: nitrogen fixation, nitrification, assimilation, denitrification, as well as the NMOX process. I told you, right, even from here also we can get the question, and we got the question also in the form of equations. What is nitrogen fixation, friends? Where the atmospheric nitrogen can be converted into NH3 or NH4 plus. Is it? Of course, yes. And what will be the nitrification, friends? Understand, everyone. Nitrification is the process where we are converting NH3, NH4 plus into more simple forms. What are more simple forms? Nitrite and nitrate. Then what is assimilation? This is the most important when compared to all. Assimilation is a process we are assimilating. Assimilating means incorporating. Understand my words. Assimilating means incorporating or utilizing this particular NO2, NO3, ammonia into what, friends? Into the amino acids. And what will be the denitrification process, friends? Where there is a conversion of NO3 back to the atmospheric nitrogen. And friends, you all people know very well that there are very few NMOX bacteria were identified. See, originally, friends, in the nitrogen cycle, what we will do, NO3, once it is completely utilized for the production of proteins and amino acid, then the remaining NO3 we are converting into the atmospheric nitrogen. Is it, friends? But I want every one of you to understand one unique statement is that, friends, in the NMOX process, there are certain bacteria. I am not saying that all the bacteria. So far, we hardly found out 5 to 6 bacteria only, which are doing this particular NMOX process, which can convert ammonia directly to the nitrogen. What, friends? Which can convert ammonia directly to the nitrogen. That is what, friends, the NMOX process. I want you to remember the every single equation as well. Now we will discuss one by one very elaboratively and try to remember maximum amount of the information from this particular concept. How much information you can remember, you have to remember that much. Okay, look into the first step. The first step is, look into this, fixing of the atmospheric nitrogen. Correct now, friends? How I am fixing the nitrogen? I am fixing the nitrogen into NH3 or NH4 plus. Correct now? So what do we call it, that particular point? Nitrogen fixation. Who will do this? Everyone can do this process? No, everyone cannot do this process. The microorganism who can do this particular process, we generally call it them as what, friends? We generally call it them as the nitrogen fixing bacteria. Do not get confused with that point, because plant don't have enzyme, animal also don't have enzyme, so only bacteria can do. Now look into the next step, friends. Once we convert the N2 into NH3 or NH4, friends, this NH3 or NH4 can be break down into more simple forms. What are more simple forms? NO2 is one simple form, NO3 is one simple form. What is the point that every student has to remember here? Understand my words. Production of NO2, understand, friends, production of NO2 and production of NO3, both is the same process. Both is called as nitrification only. There is no doubt about it. But the unique statement I want you people to remember here is that, understand, both processes are same, but the bacteria involved in the production of NO2 is different. Bacteria involved in the production of NO3 is also entirely different. I want you to remember that particular point. Now, friends, the question is very clear. Why did I converted N2 to NH3, NH4? Why did I converted NH3, NH4 into NO2, NO3? What is my ultimate goal? Ultimate goal is assimilating all these in the body. By that I can produce the proteins and amino acids. So friends, who is going to assimilate now? Of course, friends, animals cannot assimilate very faster. Who is going to assimilate now? Obviously, first assimilation by the plants. Now what the plants are doing, friends? Upon eating, so now the plants will produce the proteins, amino acids, nucleic acid, whatever they required by using this particular nitrogen. Once, this is important, from the plants who will take, animals will get. Now try to understand, friends, don't confuse with my words. Now there is a death of plant, there is a death of animals. Now friends, once there is a death of plant and animals, that dead organic matter can be decomposed by what, friends? Dead organic matter can be decomposed by the decomposers. And what the decomposers will do? Decomposers break that dead organic matter into, once again, simple forms. Carbon, oxygen, nitrogen, hydro, right now? Now understand my words. Decomposers also will take the nitrogen, how much ever they require. How much ever the nitrogen they required, they will also use it. What about the remaining nitrogen, friends? This is the important step. Remaining nitrogen can be fixed in the soil. Where the remaining nitrogen can be fixed, friends? It can be fixed in the soil in the form of ammonium. And friends, this process only we will be calling it as what process, friends? Ammonification. So we have to study nitrogen fixation, nitrification, assimilation, ammonification. Now everyone understand. Now friends, plant benefited, animal benefited, even friends decomposers are also benefited. Now what has to happen, friends? Wherever we brought the nitrogen, we have to fix it there, because we have to maintain the atmospheric balance compulsory. So what we are doing, look into this. We are sending the nitrates back. All the students pay attention. We are sending the nitrates back into the atmospheric nitrogen. This is what exactly, friends, we can call the denitrification. Okay, all the steps are important. You should not leave any step actually. Okay, now what will be the first step, friends? Among all, friends, among all, two steps are much important steps. The much important steps are nitrogen fixation and the assimilation. These two steps are much important steps, friends, where most of the times the statement related questions used to come majorly from these two steps only. Okay, let's follow. Atmospheric nitrogen can be converted into what? NH3 or NH4. Friends, what is the first statement all of you has to remember? In a given 4-marker question, they will give a statement that nitrogen fixation is a highly energy consuming process, which is absolutely true statement. You will understand, in order to fix one nitrogen molecule, friends, we have to use nearly 16 ATP molecules. It is a very highly energy consuming process. Friends, from the plus 12 onwards, we were reading, we can fix the atmospheric nitrogen in a different ways. You know, right? When the natural lightning comes, understand my words correctly, when the natural lightning comes, we can fix 5 to 8% of the nitrogen. Correct, my friends? And you know very well, industrial nitrogen fixation, you read in the plus 2. What is that process? By the Haber's process, industrial nitrogen will be fixed. But friends, we don't want to study, remember my words, we don't want to study the atmospheric nitrogen fixation. We don't want to study the industrial nitrogen fixation. What we have to study is, friends, bacterial nitrogen fixation. Who is going to mediate this particular process, friends? Bacterial nitrogen fixation can be mediated by an important enzyme, friends, called as nitrogenase enzyme complex. Please do not get confused with this particular point. Okay, what are the, and now you know very well, we are going to start this particular step. Now you know very well, we plants can't fix the nitrogen, animals can't fix the nitrogen. Now obviously, who is going to fix this particular nitrogen? Undoubtedly bacteria. Now all the students remember my words. I really don't know what you do. It's your responsibility to remember every single bacteria that I have mentioned in this particular table. You know, from the last 2 to 3 year paper, they are not raising the question from the bacteria. So I am just guessing. This time there is a great chance that you can expect the question from the bacteria also. So make sure that you remember and learn all the bacteria without fail. Okay, types of bacteria. There are two type of bacteria are there, friends. Free living nitrogen fixing bacteria, as well as, friends, symbiotic nitrogen fixing bacteria. All the students try to understand what I am talking. What is a free living nitrogen fixing bacteria and symbiotic? You all people know that, we read many times. Symbiotic means the bacteria always maintaining what kind of association? The bacteria always maintaining the mutualistic relationship with the other plant. By that, what is happening? Both are getting benefited. So understand my words correctly. The bacteria, by maintaining the symbiotic nitrogen association, they are having the ability to fix the nitrogen. That's the reason only we have given the name symbiotic nitrogen fixing bacteria. And friends, when you look into the free living nitrogen fixing bacteria, what is the point you have to understand in the free living, friends? They do not require anyone's help. What do you mean by that? They grow on their own, they develop on their own, they don't dependent on anyone. But the point is here, even though they are growing on their own, they are having the ability or capability to fix the nitrogen on its own. That is what exactly, friends, we will be calling it as the free living nitrogen fixing bacteria. Please don't forget that particular point. I am requesting you, friends, here nothing to explain. I want you people to remember compulsory. Minimum 3 to 4 times the question was asked from the bacteria. So that's why remember all. I am not marking important one also. I want you to remember all. You all people commonly know that. Especially all the leguminaceae plants. They will be associated with what kind of bacteria, friends? Obviously, they will be associated with rhizobium. Azorhizobium, Brodyrhizobium, Cynorhizobium, Photorhizobium, all this. Frankia, whatever I am marking, at least try to remember, otherwise remember all. Frankia, associated with Casuarina tree. Nostac, associated with the Gunnera. Anabina, with Ajolla. Acetobacter, with the sugarcane. These are all what bacteria? Symbiotic. Nitrogen fixing bacteria, free living bacteria, they are of different types. Some of the free living bacteria are cyanobacteria, some are aerobic, some are facultative, some are anaerobic, some are non-photosynthetic, some are photosynthetic. Don't get confused with this point. So friends, blue-green algae such as Anabina, Calothrix, Nostac, aerobic bacteria like Azospirillum, Azotobacter, Bezeranschia, and I want you to remember another example which I am writing here, Darkshia. This is also asked question, friends. Drxia. Please remember, friends. It is also aerobic free living bacteria. Aerobic free living bacteria which can fix the nitrogen in the presence of oxygen. They require oxygen. You all people know very well what is facultative. Facultative bacteria are those type of bacteria. Actually, they do not require oxygen. What, friends? Actually, they do not require oxygen, but if there is a presence of oxygen, their growth is abundant. Their growth is rapid. So that kind of bacteria we can call facultative bacteria. Example, Klebsiella. Some are anaerobic, some are non-photosynthetic, some are photosynthetic. And friends, try to understand one thing. Nostoc, Anabina, Acetobacter. What is the speciality of these three bacteria? These three are a kind of exceptional bacteria, friends. I want you to remember. What is the special quality? That is, they can fix the nitrogen symbiotically also. They can fix the nitrogen by free living also. Right. So this is the speciality of these three bacteria. Not all the bacteria can act in a both ways. Don't confuse. Only these three bacteria can act in the both ways. Friends, we are studying the first step only. The first step of nitrogen fixation, which is a very broad step. Who is fixing the nitrogen? Two type of bacteria. Now, friends, understand my words correctly from now onwards. This slide onwards, you pay attention. Focus. Friends, whether it is the free living bacteria or whether it is the symbiotic bacteria, doesn't matter. Whether it is the free living bacteria, whether it is the symbiotic bacteria, it doesn't matter. One point you have to remember, they should have certain enzymes. Among all those, what will be the most important enzyme that is required by these two type of bacteria? Nitrogenase enzyme. This nitrogenase enzyme is absent in the plant, absent in the animal also, specially present in the free living and symbiotic bacteria. This is the one. What is the function of a nitrogenase enzyme? This is the one which will convert the N2 to NH3 or NH4 plus. But friends, there is a great disadvantage with this enzyme. I want you, from now onwards, I want you to understand the concept correctly. What is the disadvantage with this enzyme? This enzyme will become completely inactive. This enzyme will become completely inactive if there is a presence of oxygen. Enzyme becomes inactive if there is a presence of oxygen. Now you can ask me why. I will tell you in the upcoming slide. Remember, subunit of the nitrogenase enzyme will be inactivated by the oxygen, because remember my words, one of the subunit of nitrogenase enzyme extremely sensitive. What friends? Extremely sensitive to the oxygen. If there is a more oxygen is available, obviously friends, the nitrogenase enzyme is getting inactivated. Please kindly do not get confused with that particular point. So friends, remember my words now. So I am a bacteria. Understand my words. Whether I am a free living bacteria, whether I am a symbiotic bacteria, what should I do now? Now I have to maintain, what I have to maintain? I have to maintain anaerobic condition while I am fixing the nitrogen. Here also most of the people will choose the wrong options. They will think that bacteria will maintain the anaerobic conditions all the time for the nitrogenase enzyme activity. That is wrong statement, because whenever majorly they want to fix the nitrogen, only during that particular time the bacteria start maintaining anaerobic intracellular conditions in their cell. So now let's see how. There are many kind of intracellular mechanisms are there, friends. One of the mechanism is heterocyst. One of the mechanism is what, friends? One of the mechanism is the heterocyst. And once again I am reminding to all of you, friends, this heterocyst is also not formed all the time. The heterocyst is also formed only a particular period of time. What is that particular period of time? Whenever bacteria wants to fix the nitrogen, only during that particular time, all the students look into that. Only that particular time the bacteria is preparing this particular heterocyst. Don't get confused with this particular point at all. Now I will tell you two statements about this particular heterocyst. What are that particular two statements? One statement is that, friends, heterocyst is extremely thick-walled. Heterocyst is extremely thick-walled. And the second and most important point everyone has to remember compulsory, the heterocyst is lack PS2. That is, heterocyst do not contain the photosystem 2. Don't confuse with that particular point. Friends, whenever the heterocyst is not having photosystem 2, you only tell me what is happening. Use the brain. If there is a no photosystem 2, what happens, friends? Photolysis of water never takes place. Yes or no, friends? If the photolysis of water is not taking place, which cannot be released, my friends, oxygen cannot be released. Yes or no? Of course, yes. If the oxygen is not releasing, then obviously it is an advantage for the nitrogenase enzyme. Suppose example, every student understand my words. Oxygen is released. Imagine oxygen is released. But do you think oxygen can diffuse into this particular heterocyst? Do you think oxygen can diffuse into this particular heterocyst? No, it is not at all possible. Why? Because this heterocyst is extremely thick-walled. Understand my words. Since it is very thick-walled, it never allows the diffusion of oxygen. See, Advantage is that first it never allow the oxygen only to release. Even though there is a release of oxygen, because of its thick wall, it never allow the oxygen inside. So remember my words. So cyanobacteria, what it is doing, it is utilizing this particular heterocyst whenever they want to fix the nitrogen. Please do not get confused with that particular point. Okay, now remember, what are the other mechanisms to maintain anaerobic conditions? All the students follow my words very correctly. Other bacteria. Look into this. Aerobic nitrogen fixing bacteria. Friends, it is really a challenging. It's really a challenging condition. Remember my words correctly. Remember, I am a bacteria. I am a bacteria. Remember, I am a bacteria. If I want to survive, I will be requiring the oxygen every single time. What, friends? If I want to survive, I will be requiring the oxygen every single time. But if there is a presence of oxygen, I can't fix the nitrogen. For my survival, I require oxygen. But if I want to fix the nitrogen, oxygen should be absent. So how I can manage? There is two examples of the aerobic nitrogen fixing bacteria. Follow me, everyone. One is called as Azotobacter. Another one, friends, we can call the Gliotheciae. What is the speciality of this particular enzyme? It maintain reduced oxygen conditions. What, friends? It maintain reduced oxygen conditions through high levels of respiration. See, friends, how different mechanism a bacteria can exhibiting. What do you mean by the word high levels of respiration? Because of the high levels of cellular respiration, more amount of oxygen is consumed. I want you to understand the depth of the concept here. More amount of the oxygen is consumed. What is happening, friends? More amount of the oxygen is consumed. Since more amount of the oxygen is getting consumed, what is the point you have to remember? Oxygen availability is low. So in the very low oxygen, bacteria start surviving because they require oxygen. Very limited oxygen is there. With only that oxygen, they will start surviving and they will start fixing the nitrogen. See, understand. Suppose if the oxygen is 100%, nitrogenase enzyme will be inactive. There is no doubt about it. But if oxygen is only 20%, of course, nitrogenase enzyme is active only. Yes or no? So understand, bacteria using only 20% of the oxygen and they are fixing the nitrogen. Understand, but this is not possible every single time. Whenever they want to fix the nitrogen only, they will increase the high levels of respiration. More oxygen is consumed, then they can fix the nitrogen. So that is how Azotobacter fix the nitrogen, even though it's aerobic. What will be the another example? Another example we can call the Gliotheciae. Friends, what's the speciality of this Gliotheciae? All the students understand. They are evolving the oxygen photosynthetically during the day, and friends, it is fixing the nitrogen during the night time. See the mechanism, which means whatever the oxygen is there, it will just release it out completely during the daytime. So oxygen level will be decreasing when it comes to the night. Understand, friends? Oxygen level will be decreasing when it comes to the night. As a result, they start fixing the nitrogen during the night. Okay. Now, facultative organism, of course, it's a great advantage. What is the specialty of facultative organism? They can grow in the absence of oxygen. They can grow more rapidly in the presence of oxygen. But what is our condition currently? Our condition is that we want to fix the nitrogen only under anaerobic condition. So what the facultative bacteria will do? They start growing in the anaerobic condition. They can fix the nitrogen. And what is the advantage with the anaerobic? Anaerobic will be always a high advantage, because if there is a oxygen, first bacteria only can't survive, right? So it's a great advantage for the anaerobic nitrogen fixing bacteria. They can survive in the absence of oxygen, and they can fix the nitrogen in the absence of oxygen only. Remembering this is not the matter. I want you people to remember the name of the bacteria, which are aerobic, which are facultative, which are non-photosynthetic, which are photosynthetic, that are having the ability to fix the nitrogen. Those things you have to remember without fail. Friends, still we are studying the first step only, nitrogen fixation. It's a very broad step. Follow my words correctly. So, establishing symbiotic relationships. Understand, friends, what is symbiosis? We all people know that, where both organisms are mutually getting benefited. Now, in a given 4-marker question, one of the statement they have asked once. What is the statement that they asked? The symbiotic relationship between the legumes and rhizobia is always obligatory. See, friends, here what is exactly the obligatory means? Obligatory means compulsory or necessary, right? Compulsory or necessary. Don't confuse. But you people just think, do you think bacteria and leguminaceae plant has to maintain the symbiotic associationship every single time? No. You have to understand, when the plant start attracting the bacteria, the question is, when the plant start attracting the bacteria, only when the condition of the nitrogen is very low in the plant. What, my friends, what I am talking, when the level of the nitrogen is extremely lower, only during that particular time only, friends, what is happening? The plant start attracting the bacteria. So what is the final conclusion? We can say that the relationship between the bacteria and leguminaceae plants is not obligatory, which means it is not necessary or not compulsory all the time. See the second line, it was clearly written. What was written? Symbionts seek out symbionts seek out one another through an elaborate exchange of signals. Only under which conditions, friends? Only under nitrogen limited condition. Understand the line. Whenever the nitrogen is limited, then only the signal is getting exchanged. We will discuss how exactly the signal is getting exchanged. Now tell me, friends, what is the ultimate goal? What, friends? What will be the ultimate goal of the symbiotic association? Friends, the ultimate goal of the symbiotic association is what, friends? The root nodule formation. This is the ultimate goal. Now, if we want to form the root nodules, all the students remember my words. If you want to form the root nodules, compulsory we require bacteria, compulsory we require the plant also, which means the genes of the plant we require, the genes of the bacteria also we required in order to form the root nodule. So friends, from the plant we will use some genes in the root nodule formation. So the plant genes we can call the nodulin genes. Don't confuse, friends. And what do we call this particular nod gene? Sorry, the genes which I think I have written wrong. I'm sorry. One second. The genes which are present in the plant we can call the nodulin genes. Friends, please don't confuse, okay? The genes which are present in the bacteria we call them as the nod gene. Sometimes instead of using the word nod gene, we use the word nod factors. In the examination, mostly they will use the word nod factors only. Okay, remember. So nod genes or nod factors are the bacterial genes required for root nodule formation. Nodulin genes are what, friends? They are the plant genes which are specific to what, friends? Nodule formation, right? Both are required. There is no doubt about it. Okay. All the students follow my words very correctly without having any confusion, friends. What are the common nod genes or the nod factors present in the bacteria? Now, I don't want you to remember the structure. But I want you to understand the composition. Means where exactly the sugars are attached. I want you to remember that without fail. Correct na? You have seen, right, in the 2019 December, this is the direct question which we got in the 4 marker. Okay. Where this particular nod factors or nod genes are present, friends, they are extremely present in the bacteria. Correct na? All the students follow me correctly. Nod factors are lipocytin oligosaccharide signaling molecule. What is that? Nod factors are lipocytin oligosaccharide. You all people know, right, friends? Chitin. We all know that chitin is actually made up of what, friends? Two different type of sugars. One type of sugar we can call it NAG. Another type of sugar we can call it as the NAM. N-acetyl glucosamine and N-acetyl muramic acid, right? And one unique statement is that, and this is the question, this is the statement they asked. They have given a statement, they have a chitin alpha 1,4 linkages. This is the wrong statement, just they asked for the 4 marker, okay? But remember all the time, friends, chitin always having the beta sugars. They are having a beta 1,4 linkages. Please don't get confused with this particular point. And one more important point you have to remember, at the second position, always there is a addition of the non-reducing sugar. This is also important, you have to remember. You people no need to remember any kind of a structure at all. If you are remembering the structure, it's waste of time. Remember that they have a chitin linked N-acetyl glucosamine backbone and a fatty acid chain on the C2 position of the non-reducing sugar. This is the point I want you people to remember all the time. Okay. This nod factors are the nod genes which are present in the bacteria. You know, right? You have seen this. This is the question asked in the 2015, asked in the 2016. In both cases 4 marker, and 2018 we got the same type of a question in the part B. So I want you to remember all the genes. The nod factors or nod genes broadly categorized into 2 types. What are those? One we can call common nod genes and another one we can call, friends, host specific nod genes. Friends, what's the major difference between the common nod gene and host specific nod gene? Name itself very clearly indicating now, friends. Common nod genes are the type of genes which are present in every single bacteria. Which means it doesn't matter whether it is the free living bacteria, doesn't matter. Whether it is the symbiotic bacteria, doesn't matter. In every single bacteria, friends, there is a presence of common nod genes. That is the statement I want you to understand. Then host specific nod genes, name itself very clearly indicating it is not at all present on every bacteria. Only a specific bacteria contain this particular gene. That's why we are calling it as the host specific. I want you to remember every single example. Common nod genes are nod A, nod B, as well as the nod C. And friends, host specific nod genes, nod P, nod Q, nod H, nod F, nod E, nod L. Right? These are all said to be what, friends? Host specific nod genes. I want you to remember. But here comes one wonderful point. What is that point? There is no doubt common nod genes and host specified nod genes are required for the nitrogen fixation. There is no doubt about it. But friends, the unique statement you want to remember is If the common node genes and the host specified node genes wants to be active, we will be requiring a specialized gene. What do we call it, that particular specialized gene, friends? We call it as the node D gene. What is that gene? Node D gene. Friends, remember my words. I have written a line. What is that line? Node D gene is the constitutively expressed gene. Friends, what do you mean by that word? What do you mean by the word constitutively expressed? The word constitutively expressed means continuously expressing in the bacterial body. And what is the speciality of this particular node D? Understand, friends, every one of you, the node D actually acting as a regulatory gene. It is acting as what, friends? It is acting as the regulatory gene, which regulate the common node gene also, which regulate the host specific node gene also. I want every student has to remember these 2 points without fail. Remember. So friends, I want you to remember the names. Okay. Last time I did not, highlighted this particular slide, but I want you to remember the only names. Very easy to remember also. Look into this. Node A, very easy, friends. Act as a acyl transferase. Node B, act as a oligosaccharide deacetylase. And node C can act as a oligosaccharide synthase. I want you to remember the names of the 3 genes and their activity. Name itself very clearly indicating and easy to remember. Acyl transferase, which is transferring or which is adding the fatty acyl chain at the second position. Chitin oligosaccharide deacetylase, which is removing the acetyl group from the terminal position. Oligosaccharide synthase, which is joining the one NAG molecule with the another NAG molecule. That is what we can call synthase. I want you people to compulsory remember. A will be acyl transferase, B will be oligosaccharide deacetylase, C will be, friends, oligosaccharide synthase. I want you to remember these 3 without fail. All the students follow my words correctly now. So friends, focus. If you understand this particular slide, the concept is crystal clear. How does the symbiotic association is taking place? It will be crystal clear to all of you if you understand this particular slide. I'll explain point by point. Remember, in the symbiotic relationship, the root has to attract what? Bacteria. The root hair has to attract the bacteria. Now every student follow my words. If the root hair wants to attract the bacteria, all the students follow my words correctly. If the root hair wants to attract the bacteria, my friends, every one of you listen my statements correctly. They are releasing certain chemicals. They are releasing certain chemicals. What do you call that particular chemicals, friends? You call that particular chemicals as chemoattractants. What do you call the chemoattractants? What are the examples of the chemoattractants? Everyone has to remember compulsory. Examples are flavonoids and betaines. What, friends? Flavonoids and betaines. These are all the 2 type of bacteria, friends, which are involved in attracting the bacteria. All the students follow my words correctly. Understand the concept. These chemicals will go and activate which gene of the bacteria? That is the question. These chemicals will go and activate node D gene. What, friends? They will go and activate node D gene in the bacteria. Now all the students follow me. Now the bacteria which is present in the soil, I will re-explain, don't worry. Now the bacteria which is present in the soil will come and attach us to the root hair. This is the difficult point. See, this is the root hair, correct na? This is the bacteria. Now question is very clear. How does the bacteria exactly attaches to the root hair? How? All the students understand the concept here. On the root hair of the bacteria, there are certain receptors are present. On the root hair of the bacteria, certain receptors are present. What do you call it, that receptors? We called it as lectin receptors. Where they are present? They are present on the root hair. So to this receptor, who will go and attaches? Bacteria attaches. Remember my statements. The attachment of the bacteria. What friends? The attachment of the bacteria to the lectin receptor present on the root hair. What do we call it that? We will be calling it as the Rica adhesion process. Don't confuse. I want you to understand the concepts very correctly. Remember my words. What the bacteria is doing? Bacteria releasing the chemoattractants. Where? Into the soil. Once the bacteria release the chemoattractants into the soil, immediately what is going to happen, friends? The chemoattractants will activate the NOD-D gene in the bacteria. Then bacteria will come and attaches to lectin receptors present on the root hair. That process only we can call Rica adhesion. Understand the concept? Okay. Now the bacteria attaches. There is no doubt about it. Now all the students follow me. What the bacteria is doing, you know friends? Bacteria causing curling of the root hair. What the bacteria is doing? Bacteria is doing curling of the root hair. Now the question is very, very clear. Why? Why the bacteria causing curling of the root hair? Bacteria causing curling of the root hair, friends, in order to enter into the root. Simple point. In order to enter into the root, it is causing the curling. Now friends, what is going to happen now? Now friends, whenever bacteria entering inside, see, look into the picture, bacteria continuously entering inside. This is called as infection thread. What? Infection thread. Every student listen my statements. Listen my statements correctly. Whenever bacteria traveling inside the root, bacteria undergo the rapid division. What friends? Bacteria undergo the rapid division continuously. It will undergo the rapid division. As a part of the division, they lose the cell wall. Remember my words correctly. It will undergo the rapid division and it lose the cell wall. Once it lose the cell wall, we call that particular structure as bacterioids or rhizoids. They are the one which fix the nitrogen. See, everything was written as a point. All the students remember, plant root releases isoflavonoid and betaines. Who are releasing? Root. Then these chemicals go and activate which gene? NOD-D gene. What the NOD-D gene will do? Even friends, this is the line directly asked in the part B question. Direct. I have already told you, right? NOD-D gene is said to be constitutively expressed gene. Is it, friends? So what the NOD-D gene will do, friends? NOD-D gene activate the transcription of other genes. You're getting my point? NOD-D gene is activating the expression of the other NOD genes. Right. So what will happen then? Remember, this is called promoter. The promoter called as what? NOD box. What do we call the promoter? NOD box. To this NOD box, who will come and attaches to the NOD box? NOD-D will come and attaches. Understand the concept. The moment NOD-D will come and attach to the NOD box get activated. Then the NOD box will be activating all the common NOD genes and activating all the host specified NOD genes. Simple point. Now I told you very clearly, right? Friends, how does now bacteria got activated? There is no doubt about it. But the bacteria wants to attach us. So bacteria bind to the lectin receptor on where, friends? On the root hair. Via which process? Via a process called as, friends, Rica adhesion process. Don't confuse with that point. And it will cause the curling of the root hair. It enter continuously. That is called infection thread. What did I tell you? bacteria multiply rapidly, lose their cell walls. That structure only we can called bacteroids. That bacteria, it's only friends going to fix the atmospheric nitrogen. How it will fix the nitrogen, we will discuss in the 11:30 a.m. class. Okay. So we will finish the entire concept in the 11:30 a.m. As usual, we will discuss all the PYQs from this particular topic specifically in the 7 p.m. class. Okay. So friends, as I told to all of you that we will see in this revision course, we will be covering 4 topics for every unit. I told you. By that only I can cover up to 55 to 60 topics. Now you people choose between one topic, between the secondary metabolites and sensory photobiology. Okay. Now if you take an average, of 10 question papers, I can say that 6 to 7 times this was asked in the exam and 4 to 5 times this was asked in the exam. That is the sensory photobiology. You people decide which topic you want me to teach. In the tomorrow class, I will finish that particular topic only. Both are important, but we have a limited time. Okay. So even you can watch the previous classes of this also. If I will take one topic according to your convenience, remaining topic, if any student is feeling difficulty, you can see the previous recorded lectures in the app. By that you can cover over there also. Now you people decide and tell me in the morning class. Okay friends, okay, that's all for now. I hope everyone understand this concept clearly. Remaining concept we will be finishing in the 11:30 a.m. class. Okay. Okay friends, that's all for now. Okay, wonderful, wonderful. Then we will go for the secondary metabolites. No problem. Okay. Okay friends, thank you all. I'll see you at sharp 11:30 a.m. Thank you all of you. Thank you so much.